The eukaryotic cells' genetic instructions are housed in the nucleus and carried out by the ribosome. The nucleus contains most of the DNA in a eukaryotic cell, and ribosomes use the information from the DNA to make protein. So the ribosome, that's the real site for the production of protein. So the nucleus contains most of the cell's gene and is usually the most conspicuous organelle, the most remarkable organelle. The nuclear envelope encloses the nucleus, separating it from the cytoplasm. And the nuclear membrane is a double membrane. Each membrane consists of a lipid bilayer. The pictures showing you the nucleus, the structure. This one showing you the structure of nucleus. So nucleus itself, it is surrounded by the membrane, double membrane, nuclear envelope. So it is composed of inner membrane and outer membrane, double membrane. And inside the nucleus, we cannot see, but there's supposed to be chromatin. Chromatin is what? Chromatin, it is composed of DNA and some protein bound together and make up chromatins. And those chromatin containing many genes, and those chromatin, when they prepare for cell division, they're gonna get coiled up, extensively coiled up, become supercoiled, become very thick and very short. Those very thick and short structure, now we cannot call them as chromatins anymore, but we call them as chromosome. And because they become very thick and become very short, now we can see very well, even when we use just plain light microscope, it can be seen very easily. That's why name is chromosome. Chromo means colored, zome means body, so it's colored body. It can be seen very well because they become very thick and very short. Now chromosome. Anyway, another structure we can see, that's the nucleolus. Nucleolus, we believe the structure of nucleolus because that's the place for the ribosome part. Ribosomes RNA parts are transcribed in that place of nucleolus very actively. That's why it looks dark. Sometimes just one nucleolus, sometimes more than one nucleoli, we can see. Ribosome, it is composed of protein and RNA. Those RNA part we call, that's the ribosomal RNA. Those some part of the ribosomal RNA, they are transcribed in nucleolus. That's why. And outside the nuclear envelope, we can see the nuclear pore. So nuclear pore, it is composed of 8 proteins surrounded up, and it can control the traffic going in through the nuclear pore or going out from inside to outside through the nuclear pore. So those, protein, yeah, the surrounding protein, they control the traffic, go in and out. The other thing, the outer membrane of nuclear envelope, it is directly connected outside into rough ER. So you can see with a blue colored rough ER, the membrane is directly connected into the outer membrane of nuclear envelope. We can see. This one showing the nuclear envelope and nuclear pore. With the image from TEM, transmission electron microscope. And each nuclear pore surrounded up by 8 specific proteins to control traffic in and out. So that the nuclear envelope has to be strong. So it is supported by the network of protein fiber here. So we can see those kind of network-like structure make up nuclear lamina, the image from TEM. So as we discussed, the nuclear pores regulate the entry and exit of molecules from the nucleus. The nucleus size of the envelope is lined by nuclear lamina, which is composed of proteins and maintains the shape of the nucleus. As we just discussed, in the nucleus, DNA is organized into discrete units called chromosome. So as I mentioned earlier, when chromatin coiled up become very thick and short, now we call this the chromosome. They are easily observed by the just plain light microscope. And each chromosome is composed of a single DNA molecule associated with proteins. And the DNA and proteins of chromosome are together called chromatin. Okay, as I mentioned, technically, chromatin, it is a structure, the loosen up structure of the DNA and protein, we call this chromatin. And when they coil up and become thick structure, can be seen very easily by light microscope. We call them as chromosome. So chromatin condenses to form discrete chromosome as a cell prepares to divide. Cell division. Ribosomes, that's the actual site for the production of protein. So those ribosomes, they can read the genetic information inside the messenger RNA to assemble amino acid into long line to produce primary structure of a protein. That's why they call ribosome as the protein factories. So ribosomes are complexes made of ribosomal RNA and protein together. Ribosomes carry out protein synthesis in two locations. One in cytosol. So when ribosome located inside the cytosol, we call those ribosomes as free ribosomes. They are freely suspended inside the cytosol, that's why. On the other hand, some ribosomes, they are attached on the outside of endoplasmic reticulum or the nuclear envelope. So those ribosomes, they are tied up on the membrane. That's why we call them as bound ribosomes. The image showing you the two types of ribosome. Free ribosomes in cytosol, and some ribosomes, they are attached on ER, endoplasmic reticulum. And each ribosome, it is composed of two subunits, large subunit and small subunit together. And those two subunits, they are composed of ribosomal RNA and protein together. Makes up ribosome. So two types of ribosome. Free ribosome in cytosol and bound ribosome. They are tied up to membrane. So that's the cross section for the ER. I mean rough ER. Yeah. And the structure of ribosome composed of large subunit and small subunit. Each subunit composed of protein and ribosomal RNA. Inside the eukaryotic cell, most of the organelle membrane and plasma membrane, sometimes they are connected together directly, physically, or sometimes they are connected indirectly to cooperate, to perform some function. So those membranes, they cooperate together. So all those involved membrane together, collectively, we call that's the endomembrane system. So, I mean, plasma membrane, that's not endo, because endo means inside. So plasma membrane, that's not endomembrane. However, by the function, plasma membrane, it is a member of endomembrane system. So the endomembrane system regulates protein traffic and perform metabolic functions in the cell. The endomembrane system consists of nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosome, vacuoles, plasma membrane. These components are either continuous or connected via transfer by vesicle. Sometimes we call those vesicles, they move around. Yeah? That's why we call sometimes transport vesicle. The endoplasmic reticulum, that's the biosynthetic factory. The endoplasmic reticulum ER accounts for more than half of the total membrane in many eukaryotic cells. The ER membrane is continuous with the nuclear envelope. So directly they are connected together. There are two distinct regions of ER. Smooth ER without ribosome attachment, and rough ER, whose surface is studded with ribosome. Sorry, cursor's gone. Sorry for that. This one showing you the smooth ER and rough ER and the outer membrane of nuclear envelope. It is directly connected into ER. And never mind for the cystine or transitional ER something, never mind. And on right hand side we can see the smooth ER and rough ER comparison. This one showing you more detailedly for the comparison between smooth ER and rough ER. And rough ER we can see some ribosome attached on membrane. Functions of smooth ER. In case of human, they are closely related to the function of our liver inside our belly. Liver, because that function for the smooth ER, they synthesize lipids, so they involved in lipids metabolism, and they metabolize the carbohydrate, especially glycogen. So glycogen, they are accumulated inside our liver and inside our muscle cell, and sometimes they're gonna hydrolyzed the glycogen inside our liver like this. Yeah. And the other thing connected with our liver, they detoxify the drug and poison by the smooth ER and stores calcium ion inside the smooth ER. So same thing. Yeah? So detoxification, it is connected with the function of our liver. Yeah? So inside our liver, we can find very proliferated smooth ER inside our liver. How about functions of rough ER? Okay, functions of rough ER is connected with ribosome, because when we call rough ER, ribosomes, they are attached on the surface of membrane of ER. Okay, so the function is connected with ribosome. Ribosome is what? Ribosome is to produce. That's the site for the production of protein. In biology, all the levels of biology, structure and function, they are always correlated together. There is no accident. There is nothing by chance. Yeah? So there are some reason. Yeah? So why those ribosomes attached on the ER? Because it is very convenient place for their function like that. So that's why function of the RAF-ER, they have bound ribosome which secrete glycoprotein, secretory protein they produce. And because it's very convenient location for that ribosome, that's why. And when they produce those secretory protein, they're gonna packaged by making transport vesicle very easily, because when they produce secretory protein, they produce protein inside the pocket of RAF-ER. I mean, after they produce inside the pocket, those pockets become a transport vesicle very easily, because that ER structure, it is not stable, but it is very dynamic structure. They can change into, they can pinch out and make up a transport vesicle very easily, containing secretory protein inside, surrounding up by the membrane. And the RAF-ER is a membrane factory for the cell. So when those ribosomes produce protein, they produce membrane protein. So after they making protein, they directly put inside the membrane. So that's why it is very convenient location for that ribosome making RAF-ER. And the Golgi apparatus, the function as a shipping and receiving center. The Golgi apparatus consists of flattened membranous sacs called cisternae. Oh, never mind, cisternae something. And the functions of the Golgi apparatus modifies the product of the ER. So when ER produce some kind of protein, they transport those products by the transport vesicle into Golgi apparatus pocket. Yeah? And inside the Golgi apparatus, they have many different enzymes. So those enzymes that modifies the product of the ER and manufactures certain macromolecules inside the Golgi apparatus pocket, and they source and packages materials into transport vesicle. And Golgi apparatus, it is composed of many different flattened pockets, and different pockets contain different combination of enzymes. So they function sorting out. Yeah, they classify the material, different material in different pocket, sorting out function they have, and sending out by packaging with transport vesicle. This one showing you the serial modification of product from ER. They receive the contents from a transport vesicle to the first pocket of Golgi apparatus. With the combination of enzyme, they're gonna get processed, and the product they're gonna wrap up by the transport protein, and they transport into the second pocket, and with the same process, they move on to the third one and fourth one, and serially, they're going to modify the product to the end. So never mind cis phase or trans phase, that's too much. Yeah. So inside the Golgi apparatus pocket, different process can go through. And the lysosome, that's the another vesicle, specific vesicle containing all the digestive enzyme inside the pocket. Then we call that the lysosome. And that lysosome actually produced from Raphael through that Golgi apparatus, then containing all digestive enzyme, then it becomes lysosome. And a lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. Lysosomal enzymes work best in acidic environment inside the lysosome. So they keep very acidic environmental condition inside the lysosome. Hydrolytic enzymes and lysosomal membranes are made by Raphael and then transferred to the Golgi apparatus for further processing, as I mentioned. Some types of cell can engulf another cell by phagocytosis. This forms a food vacuole. So like the white blood cell, sometimes they eat up bacterium, they eat up. We call that a phagocytosis. Phago means eat. Cyto means cell, so it is cell eating the bacterium. And when they eat up that bacterium, it can be surrounded by plasma membrane, the membrane. So the membrane surrounded bacterium, we call that's the food vacuole. So a lysosome fuse with the food vacuole and digest the molecule. So in the case of phagocytosis, making up food vacuole gonna fuse together with lysosome, and inside the lysosome, they contain all kinds of digestive enzyme. Those digestive enzyme, they digest those bacterium. So they're gonna get digested. So lysosome also use enzyme to recycle the cell's own organelle and macromolecules, a process called autophagy. So auto means self, phage means eat, so self-eating. So some organelle, sometimes they're gonna get bad, yeah. So sometimes mitochondria or sometimes chloroplast, they get broken and function anymore. Then what? Then those lysosomes, they approach and fuse together with those mitochondria and chloroplast and digest them. So that's why that's the self-eating autophagy we call. So this one showing you the process for the I mean not autophagy, but the cell eating, yeah, cell eating, phagocytosis. So they eat up food through the plasma membrane, and with the lysosome, they're gonna get digested. Yeah, this one showing you the autophagy, self-eating. So some organelle, when they get damaged, those lysosomes eat up and digest them and make recycling and make cleaning inside the cell. In vacuole, they function diverse maintenance compartment. And vacuoles are large vesicles derived from the ER and Golgi apparatus. So vacuole, we can find in plant cell. In young plant cell, they have many small vacuole, but when they grow up, when cells getting bigger and older, those small vacuole, they fuse together and become a one large one. We call this the central vacuole. And those vacuole depends on different types of cell. They perform different function. Yeah? So they perform a variety of functions in different kinds of cell. Some types of cell, like white blood cell or some protist cell, they can go through phagocytosis, making up food vacuole. So that's the food vacuole. And contractile vacuole we can find in amoeba cell. Those amoeba cells, they have vacuole, so into the vacuole they can pumping in water and pumping in water in and out, and it can swell out, and it can be shrink, swell out, shrink. It can repeat those actions. Using that action, they can move their cell around, move around the environment. That's the contractile vacuole. And central vacuole, as I mentioned, in mature old plant cell, small plant vacuole, they fuse together, making up one large vacuole, then we call that's the central vacuole. Found in many mature plant cells, hold organic compounds and water. So plant cell, it takes about large space for the central vacuole, because especially for the plant cell, the water is very important, especially when cell growing up, they contain a lot of water inside the central vacuole and make pressure, then push outside to expand the cell size to grow up. So we can find the central vacuole, a lot of space it takes, and chloroplast and nucleus and cytosol, and most of the contents inside the central vacuole, that's the water, and water soluble molecule inside. So as we discussed already, the endomembrane system inside the eukaryotic cell, most of the organelles, membrane and plasma membrane together, they cooperate together for that metabolic function inside the cell. And all those membrane together, collectively, we call that's the endomembrane system. So the endomembrane system is a complex and dynamic player in the cell's compartmental organization. So most of the organelles membrane, they are the member for the endomembrane system. However, there are two exceptions. So one, mitochondrion, the other one is chloroplast. And those mitochondrion and chloroplast, they are not the member of endomembrane system. So they don't exchange membrane with the endomembrane system members. So between or among the endomembrane system, sometimes physically they can be connected, or sometimes by the function they are all connected together. It's showing you just briefly for the endomembrane system.